use finally. So we will use antifairs.org URL to add the notation. So this is true for any resources we use. For example, if you want to use Mammo, then I'm going to use this Mammo. It's not easy for me to select. I have to select those things. That's fine. If I have to use Mammo, I'm going to use Mammo to do 4.6, which corresponds to the Mammo term for ordinary differential equation. That's a mistake there. Yeah, it has to be underscore. That I need to fix it. Yeah. Okay. So the right one is with underscore Mammo. This is the ID, and then you pass it on to antifairs.org. It's going to resolve it to the right page. Now you have this information that this is not an differential equation. So this is good because for the search engine, when it has to search, when you give it a term, it's going to search, go to this, and then gets on differential equation term, but also it looks into a synonym, so you don't have to provide lots and lots of information. If this is also true for a gene, so it says also alternate label, so you can pick up all the information. So if I use this ontology, if someone searches for ODE or someone searches for ordinary differential equation, it should find the same model basically. The way we do is, of course, the biomodel is going to fetch information from this ontology. It's going to get all the alternative labels. It's going to get the definition, and it's going to get the name, and then put them in index them so that when someone searches it, everything is being considered. So if I have the ontology term for cancer, then it will have synonym as tumor. So someone searches for tumor, still you should be able to find the same model. So the way it works is that the biomodel's infrastructure is going to fetch the information from the ontology term, get all the synonyms and definitions, and then let the user find the model easily based on all the terms one can potentially search. So we use antifairs.org as a way to annotate, and we use ontology terms to add annotation instead of writing free text. Okay, so just to give a quick over here, so we have here model annotation. We can discuss here and agree what is the minimum we need to do for our cellular pot models or multicellular models in general, because it's very generic. And then we have this model level annotation. We have species, and which could be different components, entities. And then we have reactions. For this we use reactome, for example, and we have parameters, and we use systems biology ontology. And this can be, for example, rate constant, catalytic constant, Michaelis-Menten constant, so on and so forth. I know that in the models which we are going to talk today, we have different things like which are physics-based concepts, forces and tension and whatnot. So we need to contact between cells. So we need to find the right terms so that we can include in our list. If these terms are missing, we could potentially consider adding them to the systems biology ontology. So because that's something which we have control over with us. Or if there's a better ontology which is already existing, then we can use that. That should be fine too. Okay, so I think I have given enough introduction. Maybe we can see what could be done next. So what do you propose? How should we proceed with this session? So should we pick up a model and then try to see what's the best way to annotate the model? What is the minimum we think should be there for that model?
I could show an example of a Morpheus ML model that is already at Biomodels database of how we were hiding some of these annotations in our current Morpheus ML structure. And you could say that's completely useless how we do it, and we shouldn't do it, and wait rather for the proper way of getting these RDF bags into the language, or whether we should still do it. If we should still do it, then we have some other models that we could live annotate in this way and maybe upload to biomodels.
Yeah, I mean, so RDF bags are one way to do that. I mean, it's totally up to us to decide as a community in FAIRSPACE what is the best way to do that. If you, I mean, we can put this RDF even in Excel or CSV file, right? Basically, you need to find the qualifier and the ontology, and then next one will be the URL interface log URL, and that's going to be good enough as well. So it's going to have the information that we need, and then we just need to tell the BioMods to read from that instead of reading.
So I have a question. Maybe I'm missing something very basic, which is I understand how to annotate the entire simulation. But if you're annotating model simulation components, how do you maintain the connection between the annotation and the component itself, unless you actually put them as decorators, as comments, for example, in the code? Because my understanding was that what you really wanted in biomodels was a separate file or a header file that was like it was an SBML file that would be something that could be parsed by your tools.
Yeah.
Whereas if we have CompuCell Python and we have decorators living in the Python, then you're going to have some way of extracting that, and presumably you'd want to maintain somehow the connection between, say, a Python object and its definition. And there also is a question which is less of an issue with SBML models, because the networks are a very limited class of model in some sense, which is how do you annotate processes, right? In a network, essentially all you have are time changes, all you have are rates and events. But in the kinds of models that we're talking about here, you have things like adhesion, cell division, which are temporal processes. I'm not saying that you want to annotate the time, but you at least have code that implements those things, and you have to tag those then to the thing that they're doing functionally. And so I'm not saying that this is an impossible thing to do, and I think any annotation is good. I just wanted to understand. I mean, I thought we were going to do the simple one, which is basically adding author information, comment information, and so on at the header of the file, which already is more than we do. But I'm interested in thinking about these harder problems as well. I don't mean to derail the conversation. I just think it's a challenging and interesting question.
No, I agree. Yeah, I think that's very interesting too. Yeah.
And especially because in CompuCell, you have a lot of flexibility and you can come up with different ways to implement the same process. But yet you can actually also, as an user documentation, as you now make comments and say the following text or command block is meant to do this and this. At the same way, you could extend this comment by referencing.
Yeah.
And I mean,
Probably
I think CompuCell would do better if we gave people less flexibility. In other words, say the conversion from Monte Carlo steps to minutes is the term. We will use this variable name, this parameter name, and just say that's what it is. And then you have a tag. Then it's easy enough to make an XML tag that's just that. And to make it a little bit more the way you've done it, I think is probably very desirable. But that's a little bit different from the kind of annotation we're talking about here. But they're not unrelated, because once you decide on those conversions, for example, the names of those conversions, then you could give them tags. And since Chi-Chi's here, I should say that all of these things are not just CompuCell or Morpheus dependent. They're sort of general problems for agent-based models, not just for ours.
Yeah. Anyway, so let me show you quickly how it is added to SBML model.
I mean, I guess the other thing one has to think about a little bit looking at this,
yeah,
is right. SBML is never meant to be human readable.
Yep.
The Morpheus ML is sort of in between. CompuCellML and CompuCell Python are meant to be read by people. And so as you add more annotation, then it becomes more and more essential to have a browser, an editor tool, because you don't want to be reading what you just showed, right? That's not human readable.
Yeah.
So there's a bit of a tension between embedding the annotation and making the code human readable. There are a lot of benefits to having it fully annotated and using a viewer. But then you lock it to a particular tool or set of tools. Whereas if it's human readable code, you could write CompuCell code in Word, in Notepad, in any editor. And so it's something. Again, I'm not saying it shouldn't be something we do, but it is something that we've struggled with. This is one of the reasons we don't have an annotation standard.
Well,
is how to square them.
Maybe instead of a browser viewer for this, you just need a simple converter. It seems like just some parsing of the document could pull out the important information and display it for you. So it's still in text, it's not anything really fancy, but just some smart parsing could do the trick. Of course, you don't want to parse it and then lose the annotations. You want to keep the annotations. So you want to be careful how you handle that.
But eventually, I think this hits many different platforms. So we have a fully declarative language, but if you are programming a piece of code, you also want to annotate that. And of course, you can plug a lot of documentation tags in there, but that becomes utterly ugly. So maybe it's really worth thinking of how James initially suggested, to split them off, to just tag your implementation with the kind of decorators that are really identifiers, and then have an extra file which can then have an editor, and everyone can use it and refer to these tags. That appears a little bit more convenient on the side of human to preserve human readability, and also use this more structured RDF tags, which is
That's a great idea. I mean, if you have at the end of each line of your CompuCell or Morpheus code something that says in the annotation file, this is annotation number 677.
Yeah.
No, I rather would do the other way around. I would just try to put a unique tag for what is going on here. Say, this is my cell type ABC and cell division going on. And then you can refer to an annotation tag that uses this identifier and says, I'm the documentation for this identifier. And then even if you move things around, these identifiers will be kept.
But maybe I didn't express myself. I think we're saying the same thing.
Okay.
In the code part of it, your comments have a tag, a name, a number, or a unique identifier that ties to a document that has all of the expanded tags.
So
you got to be able to go both directions.
I'm curious about the function of this document. So this is separate from the model. This is separate from the SBML. It's like a document for the different types within your code base.
The document could be an SBML. The thing is that the annotation lives in a file that's separated from everything else. So it's not mixed in with the code.
Yeah.
But it's like so you use CompuCell 3D. It has annotations, decorators inside the code. Some of those are registered, and those are pulled out into a separate document so that you can share it and tell everyone. Hey,
well, I wouldn't say it's pulled out. I would think that actually what would happen is if I annotate something in CompuCell, my fantasy is it pops up a window. I put in my annotation. it puts that annotation into a separate document, which is some long, hard to read XML document. And then in the CompuCell code, it just says hash 666663, which then allows me to always pull up. And then if I click on that, it goes to the reference document, pulls it up, and makes it readable to me.
Okay, so this is the function is so that when you're going through the code in CompuCell 3D, you can identify what each part's supposed to do. Okay, the document isn't like declaring what was required for your particular simulation. It's actually just for reference from within the code base.
Oh, it could certainly also have that kind of information in it.
Yeah.
But that wouldn't necessarily be tagged to a particular line of code. That would be talking about the code as a whole or some section of the code.
Yeah.
Now, I don't know how you make sure how you would identify if you just have an index, a tag in the code itself.
Mm-hmm.
How you identify the scope of the tag. In other words, if you want to annotate a block of code versus one line of code, how do you indicate what and then you basically reinvent all the mess that we're trying to not have to show, right?
Yeah.
So it's a little bit tricky. Anyway, I don't want to derail. I should let Sharif continue to demonstrate, because I think what he's showing.
I think it's fine. I think it's important that we have this discussion. So in SBML, we have meta ID for everything we define within SBML, right? So that's why within that you either directly create annotation bag and add annotation inside it for each of them, or even the combined archive specification suggests that you create a separate annotation file where you put all the annotation, but then refer the meta ID of the component within the SBML. So that's fine. But then when it comes to your code, like what you have in C3D, how do you then tag different part of the code and then say this is annotation that should go here, even if you create a separate file for annotation? That I think, I don't know, we need to maybe agree on something that would be a way forward.
So I also see this flexibility that Sharif mentioned. You could tag the same object in the model with various information. You could say this is part of something else. At the same time, you can say this is not this thing and it takes part in something. So you can probably assign many relationships. It's an 1-to-n somehow relation for every model component. And that's why probably this separation of the annotation from the code itself makes it more readable. Then now if you find out, okay, I have so many things, I know so many things about my model, and I want to share all of those, you just end up with lots of your relationship and references. At the same time, I think all the code documentation that is going on, if you write C++ code plain, you also have tools to help you annotate your code and keep this transparent, and you can open and close the annotations in such code documentation tools. That could also work, that it's handled by some IDE infrastructure, and you don't need to do it yourself and invent this link between the two files.
So I just want to be clarified. So we are talking about annotation to model, or we are talking about annotation to the code for the software too?
In this case, we have both options. So we have Morpheus ML, which is a model description, not code yet. And there it may be easier to assign to components in this model description these relationships to references. But you have many multicellular simulators that contain code pieces and therefore are more flexible. And you want to do the same annotation for those codes. And then CompuCell 3D is this example here. So you want to do both.
For the code, for example, the C++ code, you will do the annotation. At least there are some software tools you can do the annotation, and you can extract those information. For example, you can use the oxygen, and then you can put the notation, the node commands into your coding, and then you can extract into the documentation like the HTML. That's really the human readable, and you can see like the API.
Yeah, the Toxygen framework, that's also something that we are using.
Yeah.
Should carry over to Python code as well.
I mean, the code itself being the actual, you know, the thing that's being annotated. It's like you're going to share people have to read the code. I'm curious if it can also relate to particular simulation results, because often with agent-based modeling, you don't use all of your parts of your code base. You use actually a very few pieces. So if you could, you know, I used chemotaxis, I used this type of adhesion, you'd want to be able to pull that out, save that somewhere else, and especially alongside a publication, you know, what pieces of the actual code base were used for this result. I also think like little kind of weird little things like the transformations between data, you know, how you go from like the perimeter discussion we talked about yesterday. You want to also possibly annotate that, right? Like we used the actual perimeter versus the idealized perimeter. You want to be able to annotate that, save that somewhere, and share it with your publication in your publication for reproducibility. Especially if later on the code base changes, you know, you update it, you add new functions. It'd be nice to have a record of how it was before.
True, and in the model description domain, like Morpheus ML, we do have, of course, then tags for that. So if you choose to normalize your surfaces, then you will have somewhere in this tree surfaces, scaling, and then an attribute normalized or unscaled or size scaled or whatever.
But I mean, I think what you're talking about, though, actually, with our experiences we were talking about yesterday, illustrates some of the complication here, which is that actually, if you're using the Morpheus editors, it's easy to see these things. But if I just have the code, your ML, trying to figure out what it does is very hard. Walking that tree by hand and trying to extract it is hard. We find this a lot when we're doing network modeling. Everybody I work with prefers to use antimony because it's very simple and it's human readable, rather than, and you don't need an editor for it. You just type it. Now Copazi has wonderful features. Cell Designer has wonderful features, and so depending on what you're trying to do, that's different. These are different things. But there is this tension, I think, between, you know, how much intermediation is required between the and let's not call it code, let's call it model specification, because it's true that we use Python for model specification, but it's still not actually, but the code is actually in C++. The Python is just coordinating C++. So it's more like the XML specs that in fact, most of our Python translates directly into XML specs that Morpheus has. They implement the same function.
So Sharif, do you want to show another example that you have open here? Else you could show one of the Morpheus ML models that do have a preliminary annotation included, and we could discuss. Or just say something about the example that you have here, if you want.
Yes, I just wanted to quickly point to the example here. I mean, to show that. So you within this BML, you have this annotation bag where you add everything. Authors who basically curate the model, then this, and then all the information which we discussed earlier, they all go within the annotation bag. Then you open annotation bag and then you can close it. So this doesn't have to be in SBML. This is not part of SBML. So this can be within any XML. Right.
I got a question. Is there some scheme available for that RDF? Yeah. So it just has an XML namespace, yeah? So it's known what
basically
can be.
You have to, the namespace are basically the combined qualifiers, right? So you need to use this BQ by all, BQ modulus, and then you have RDF back, and then you put the link.
And the RDF is just the RDF. It's just text.
It just takes RDF,
And it's up to you to understand what that might mean.
Yeah. So this, if I have this in this structure, then Copasi will read this and show me in a nice format where I can edit in the editor. I can add the ontologies and what directly. And then I can get this information, pass this through, I can get the information as I need. So I add
online.
Sorry, what did you say?
Is there some online database to crawl to the available identifiers, or is it just manually to be done in a browser?
I mean, there are APIs to do that automatically. So that's how once you have all these information, then you can fetch them. An example here is, so if you go to this model and there's a new tab called components tab here. If we go there, it will all those pieces. So you see S7, S8, that's the name that the person is using the model, how he defined the variable, and then these are the annotations. So this means inactive receptor, kinase, and mitogen activate map kinase 3, and so on. So you have all this protein. This information is extracted from the UniProt based on the annotation and displayed here, resolved, and you have reactions and they're displayed here. So you can extract this information from the model, and then because you can pass through the model, you get the information and then display here nicely. It can be, I mean, it's a web our web interface can be your editor, Morpheus editor or CC3D editor, where you show this nicely and allow user to edit this like the way you do editing in, for example, Copasi. And somehow you can, I mean, it depends what you want to do. So if you want it to be readable for some user who reads the code, or he opens the code and reads them, or is it if you want them to be readable only through your interface. So if you want it to be readable through your interface, that's fine. Then they load the model and you can store in whatever format you wish, RDF, and then the reader is going to show that nicely, read and pass it and show that nicely in the interface, the editor, and where you can edit things as well. And then that's totally fine. It will be readable only through the editor, the interface you build in Morpheus or CC3D. I mean, so if we know what format it is going to be, then from biomodel side, we need to then, I mean, if the whole point of here this meeting, I think we need to agree on a standard or a way to add this annotation. So once we agree on that, then biomodels can learn to read that, pass through that, and then index it, make it searchable, findable, and then in your case, you make it visualize these annotation in your interface GUI.
Yep.
That's what I would quickly wanted to show here. Okay, let's go to some of the Morpheus models.
Before, let's discuss James' questions.
Oh yes, okay. Sorry, I didn't see the chat. Yes,
in the chat. So I think indeed you in the network models you basically have species and reactions that you parse, and we would have much more diverse behaviors and cell types and arbitrary objects.
Yeah. Okay, let me just open an example in Copasi and show you how that looks like. I think that would be a bit more clear. I
mean, something that SBML didn't originally have was the concept of there could be more than one cell in the world. And so the fact that you could have 5,000 instances of the same species in different situations. And so this this difference between a cell type and an instance of a cell, for example, are pretty fundamental and are not encompassed in what you've got there. That's why I'm not saying it's wrong, but it's something you have to think about. Doing what you described for cell type is interesting. Doing it for an instance of a cell is also interesting. And of course, people are trying to build MLs for that. But the annotation is maybe.
Actually, this part is part of another breakout session for the rule-based modeling. We touch about the compartment type, species type. That's why we have several breakout sessions for that part.
I mean,
I just point out, yeah.
Right. I mean, we don't, in our models, the instances are generated dynamically. So you can't annotate the instance. You can, if you save the model output, you can annotate the instance, but you can't annotate the instance in the model specification because you don't have the models, but you don't have instances until the model is run.
But the instance corresponds to the type declaration. So you'd have like the cell types that are allowed in that simulation. You'd list them. Of course, you wouldn't list like all 1,000 of them in the type declaration, right? The instance declaration would be much longer in some ways because you'd have to list every single variable and the value it takes, whereas the type is compact, but also you have additional annotations maybe along in the type.
So we were also thinking of the type annotation first, yeah.
Right. Yeah. I agree. I think that's sensible. In both Morpheus and CompuCell, I mean, and also in SBML, you can overload the types though. If you have, right, the type is fundamentally a template.
Yeah.
And so the thing you annotate, I mean, again, it's not that these annotations have to be perfect. Anything is better than where we are now. So I'm not trying to say we have to do everything. So, okay, that's fine. That's clear then. Yeah, go ahead.
Okay, so just as you asked, here's a SBML, which is how to read. Now, very important to Copasi, you can see if you go to annotation tab, you can see them nicely organized here. So you have the identifiers, you have the tissues, gene ontology, all the information here. So if you want to, let's say, let me get a browser here. Know what this is. For example, it has gene ontology, GO. I need to click on that. It's going to open me the. First, go to the identifier org, and it's going to then redirect to the right. Oh, sorry. Maybe something else. Maybe.
Yeah, that's why you have to test before.
Okay, so here, so let's say I click on this memo, and then it takes me to the memo ODE example which I showed earlier. So you have this annotation nicely tabulated here, but you can choose the qualifier. If you want to add a new term, let's say I'm going to click here, choose has property or has taxon, and then I'm going to add more and more annotations here. Has version or is property of, for example, occurs in. I'm going to choose occurs in. I'm going to choose cell ontology here.
I'm sort of surprised that it doesn't automatically have a third column that gives a short definition, because what you see there is not very informative. If you click on it if you have to click on it to see what the hell it means, that's not that useful. You'd like to have a column which says go term such and such is ODE, go term such and such is B, go term such and such is ATP.
Yeah. Yes, that's totally correct. That's why okay, so let me show you one thing. So all these IDEs are somehow in this resolved here, right? So we have has property, mammo is on oriental equation, gene ontology into immune response, hemopoiesis, bone marrow occurs in bone marrow. So all these are resolved here, and what we're doing here is what you asked, right? So maybe the editor Copasi should also do that and then show it in the next column, the names. So that would be really nice to have that. Agree?
Because basically the Copasi editor makes it impossible to change anything, right? I have no idea if I opened this and I wouldn't know what I was working with, right?
Yes, you wouldn't know. You need to click on that and see,
Right.
or you need it won't show you automatically here. That would be a really nice feature. So that's what we do this here in our browser in our infrastructure and show everything. And then if you go to the components tab for another model, I need to put that, for example, here, and it's going to show what these species are basically. Let me show again here. If I go to this model and then go to biochemical and then the species, and it's going to show me for each species, each component, what they are in the annotation. And it's going to say is version of NCID and some ID. I do not know what that is. Unless it's properly resolved and shown in the next set. I need to click on this to then know what thing is. So maybe if I click on this, I know that this is a mature B-lympho site, but without that, I don't know. So I think it's a good point. Would be nice if when you build your editors, would be nice if you consider this feature for Copasi, for CC3D and Morpheus. It's a bit of extra work, but yeah, that could be a way to do that.
Sure. I have a question. So for the biomodel, is the biomodel provide functionality to download all the data from the biomodel? The reason is I know biomodel is supported by the EBI is relatively stable, but anything is online and always, no matter how stable current for resources, is always there's some risk for funding. But if you have the provide service downloadable the data, and the user always can have the snapshot in certain time point, they can maintain the link to the local, they can have local data somehow. Like the UniPro, I think they have the functionality. You can download all the data about protein. And I wonder the biomodel also, if biomodel also have that feature or service.
So we had this service that we kind of put everything together in an FTP server, release once in a while. We used to do it in the past, but we stopped doing it. But now we have APIs, which means you can download anytime. So that's API. If you call that, it's going to give you all the curated models, and that's API. If you call it, it's going to download all the models locally. So you can do it anytime you want. You will get the most updated version. So that's then we didn't do the FTP storing of releasing. But yeah,
the user have to have a special request. It's not regular released by the.
Yeah, there's no. Yeah, there's no. You can download any. Yeah, you have to request using the API. So basically, get them as you want. But there is no regular release of all the models together. We did in the past. Maybe if there are more people asking for this, then we can definitely do that. I think shouldn't be a difficult task for us to do. Yeah.
Okay, thanks.
Okay.
So shall we to go to biomodels again and then open one of the Morpheus ML models?
Okay, sure. Let's go to biomodels. Search here.
The second one of the list.
Yeah, this one.
Exactly, because there we put in some metadata.
That's good. Let me go here.
Right. If you just preview the XML file. So here we have this details tag in the description just at top. If you scroll up again,
Yeah.
That has just our human readable stuff, like we put in units and so on, just in plain text. And then we have these identifiers to, like the MAMO saying, now this is a partial differential equation model and so on, just for us to save the information for a time when we know how to properly transfer it to you.
Okay. All right, that's good. That's good. Yeah, I think because it's in a, okay. If you do that in SBML, then we will be able to read. Or if you put in if you create for example here, a bag called annotation somewhere, I don't know where, and then put all this information within that annotation bag, then we should be able to adapt the code we use to read, parse SBML, and then say pass this through, and then just take the anything within the annotation bag and then show in Biomedical. That's possible.
And if you scroll down a bit, I think we have some field variables defined, and they also have these annotations. I think it was more in the middle. If you go up again, I think those lines that start in the beginning, exactly those. Yeah. So when we have variables like psmat2, then it links to the two different UniProt and Pato resources that identify in which species and which molecule it is.
Yeah.
But that's more the human interface, right? So we have just a plain text field that opens in the editor. When we right click on the species, then we can put in any user annotation, something like to do, I have to clean up this part, or I can put in the copy paste URL that we have here.
Yeah. So,
and I understand that we have to basically go through the whole model. And now in this case, only we annotated the variables, but there are additional aspects here, like the diffusion equation, then the decay term, and so on, all the reactions that we have down here, which are not annotated. So the model is far from being annotated. But basically, we would scatter a lot of these annotations and then RDF bags now across the whole XML tree.
Yes, you would. Yeah. I mean, we need to decide. Decide as a group what's the best way forward. If you do that, if you put within RDF bag within each of these elements, then that's totally fine. Fair. We know what corresponds to what, and we need to. I mean, from our side, we need to pass this, show this, index it, make it more findable. Because if you go to Biomedical, you can search based on gene ontology terms and find a model of not signaling and whatnot. Yeah, you can use that as keyword search. And if you then want to implement these in your editor, then of course you need to find a way to pass that. So if you agree on something common, then we can all do the same in the same way. Then we can all build our own parsers to deal with that, or even share some of these parsers as needed.
I think Jörn will at some point extend this annotation field so that you can click and say, now I want to include an RDF bag, and then it will mix the plain text with an RDF structure.
Yeah, when we have full support, of course, we will add that. But I think so far you could easily also dump the RDF text right in the annotation. So Morpheus will ignore all these details and just skip that. Because that's the way it's implemented. So I think we should test that. But as far as I know, you can dump everything right there. And it will still work.
So
the editor will not show it, but
exactly. Paste this multi-line RDF bag into the text field in the editor. Yeah, and it will maintain it in the XML.
Not in the editor. That will fail.
Okay. Right, so it will not write it. Okay, I will have to do it in a text editor, and then when I open it in the Morpheus GUI, it will just ignore the content of the annotation text.
Exactly, that's it.
Mm-hmm.
So you add in all your RDF within this bag, this annotation bag, and then in the Morpheus for now you ignore it, but later when you build an editor that specifically get this information and shows them next to your all the buttons and whatnot.
If that has biomodels.
So yeah, we will read this in Biomedical specifically. Yes, we will read this and then we will index this, and we will display this in our web interface. Nicely.
Then we should try this, Beat. We will first make a normal Morpheus ML file, make the annotations by human GUI editor and then go through the text editor and just paste the RDF bag structures around all our identifiers links. And then try uploading this to biomodels and see what is done.
Okay, but biomodel is not going to read it right away because it will look at the format, and because it's not SBML, it will say I do not know what this format is, and it's going to ignore it. But we need to tell, okay, even it knows it's a Morpheus. It knows that it's a Morpheus format. So then we should tell that if it's Morpheus format, then just go and pass and get the annotation bags and get the information from there. So that needs to be implemented. So if we say that's the way you're going to move forward with, then Tung can basically implement that. So right now it can recognize CC3D format as well. So if you submit a CC3DML, it will know this is CC3DML, and it will know it's XML when looking into the into this code, and it just stops there. It doesn't do anything further. It knows this is this, and then that's it. However, if you say go one step further and get all the annotation bags, pass the information, index them, everything that's doable, basically recycling some of the codes we have for SBML, and then they should be able to do that.
Very good. So we will test whether our manually extended Morpheus ML file can still be opened in the GUI without crashing the GUI with the extra text in it. If this is like this, then we will, for the time being, do this manual extension by RDF bags.
Yes.
And then you can see how you can extend your importer when it recognizes Morpheus ML, that it also crawls into the bags.
Yes, I think if you can, I mean, if you can do one example for Morpheus and one example for CC3D, and then we can just try to implement things from our side. Unfortunately, Tung isn't here today. He could work that out and see if that is going to, that's possible and easily readable, and then we can be indexed. Then all the annotations you do will be easily findable in the biomodels search.
Okay, that's good stuff.
There's a few comments. Sorry,
we'll try. This will try, yes.
Okay, good. I think, I mean, we can even try now if you want to do that as well as an exercise to start with. So Aaron said he has been the schema of everybody. So Aaron, do you want to say it loud what you shared here? Okay. Aaron, do you want to comment about your chat?
Oh, let's hold on. The chat I had on the vivarium schema.
Yes.
If we have time for it, I could show it. It's not exactly in. So here I'm not I haven't introduced vivarium yet, but it's here I can actually send a that there's a link there to vivarium if anyone's interested in it. It's a kind of a software for integrative multi-scale simulations, not specific to any type of simulation, but more of like a integrator for many different pieces that you could put together. We used a bigraph format, as you can see here. So if you scroll all the way to the bottom, the whole cell model of E. coli was actually what it was originally developed for. You can see here we have different processes declared, chromosome structure. Each process has ports, and then wires connect them to different state variables. And so you can see here the wires go from a port name, such as fragment base, to a path in this hierarchy. In this case, fragment base goes to the bulk state, which is at just the top level. So the tuple that it maps to is a path, kind of like into a directory structure. You can see active replisomes goes top level unique down one to active ribosome. So if you scroll to the bottom of this and click on the link, it'll show you the simulation wiring diagram. And kind of zoom up on this. So you have the rectangles are processes. Each of these are kind of like the functions that drive a simulation. Ports you can see with the black dots. These are the inputs or outputs of the process, and the wires connect them to some state variable within a hierarchy. This can be submitted. So chromosome replication in this case was an ABM. Chromosome structure as well, kind of operating on some of the shared variables. If you scroll down towards the bottom, you have complexation, equilibrium, polypeptide elongation is also a kind of ABM. Scroll up a little bit. So here you have complexation is a stochastic algorithm. Equilibrium is a steady state algorithm. Each of these could be curated and annotated to different degrees. You just pull that piece of code, connect the ports up, and run the whole simulation like that. So we can take the entire E. coli model, and we've done this. You can take that and plug that into a super simulation, which has an environment, and I can share a link to that work as well. But anyways, it's a nice format to kind of share save to some file, like an exchange format, which processes you're using, how they're connected to variables. That can be taken in and simulated as is. I'll share a few more links so you can browse if you're interested more, and definitely feel free to contact me if you want to talk about it.
So the annotation is in the JSON.
It's a kind of like a condensed. It's JSON. The format you see there could be expanded to proper JSON, but kind of in the spirit of actually what James had brought up, I wanted to make it even more human readable, so it's parsed out to just the information you need, rather than like types and you can directly read it. It's complete. It is actually complete information. So what I showed you there is sufficient to specify an entire simulation. That is, if you go to the processes also have some more information. So you actually, it has the process name. You actually have to go to the process, ask what types its ports are, and then those have to match the wires that you've declared. So if you know where the process is stored plus this information, that together is sufficient for a simulation.
From your JSON annotation to graph, is the implementation is the command from JSON can generate the graph, or is your
and even in that notebook that I shared, there's a method. It's a separate library, not connected to Vivarium. That just takes the JSON format, gives you the
that's nice.
The graph is up. Yeah. You also have a validation library, also separate from Vivarium, so you can take a schema and an instance, validate that all the types are correct and matching, that you don't have like a port hanging that's not connected to anything. It can give you some recommendations too about like, hey, this process is disconnected, or you can try to connect an integer type to a float type. Please try to get this is not a valid simulation, etc.
Yep. Good. Thanks. I think quite interesting. James, I think he's commented that we could try to replicate one of the Morpheus models in CC3D. Okay, yeah, sure. We can try to do that as well. Maybe
that might have been better yesterday than today, but I think it would still be a good thing for us to do while we've got our attention.
Yeah. So I think we could start working on that and then continue to do this and maybe bring this tomorrow. I don't think we will be able to complete that if we start now. We have only a few minutes left for the meeting. There's no harm starting to work on that. and also try to add the annotation, as you said, for maybe let's pick up one model, maybe Lorenzo's model or one of the Morpheus model. So then we can add this annotation RDFs and then see if we can implement the support from our side. And if you tell that this is not going to crash CC3D Morpheus, then it's great. And then you can think how you can pass this for your own editor in future. But from our side, we can see what can be done.
So I have a question, Sharif.
Yes.
Isn't all text, doesn't XML treat all text that's not between tag pairs as not as comment?
So again, I didn't. Can you repeat the question again?
I thought by this that anything that's not inside a tag in XML is a comment.
No. You cannot do that. You need to add. Okay, if you want to put a free text in it, you need to create within that something called notes. Notes, the free text / notes. I think it's going to take that as free text.
No, but I'm asking you so the opposite question. In other words, if I took a Python file and I put in comments tag pairs,
okay,
would I be able to just process that and have that everything that's not in a tag pair be ignored?
I mean, at the moment, Biomedical doesn't do that. I mean, if you want to do that, then why not? You can try that.
Because in that case, you just put hash and then open annotation, close annotation tag.
Yeah.
In any line of your Python.
Yeah.
And then you don't have to do anything.
Yeah, but
just to intercept here, it could be a good option because Biomedical surely will not parse anything but XML. Because
right. So just ignore all the free text.
That's another infrastructure. But like I think I already mentioned, there are tools like Doxygen, if you have C++ or other tools for using Python to extract parts of the documentation. And you can really tag these and say, okay, this is just for my annotation. And then you draw all these kind of code snippets out and put them in the separate XML. So that is feasible, just like a multi-processing line to extract these in source text, if this is what you wanted to achieve.
I think there a difference between the annotation for models and the commands for the code. So the annotation for model, I think, will be considered part of the data, and then that's why we do not really, even in the JSON or XML format, we do not put their native command format. We can by using certain tag and by adding annotation. This is part of the data. And then for the source code, even for XML itself, consider as source code if that consideration, and then those things they try to for the people to, especially developer, to understand certain part of code, and those things will be ignored by the compiler or some program, the library. But when you read data, so there I think there's some difference. So for the commands in the source code, its intention to be ignored by certain, for example, compiler or something, but developer will read, and the annotation to the model is really, we want to, is part of data, so we can process those annotation when we display or exchange the models.
okay. So
by the way, I got some good news from Morpheus. So you can just paste the SBML-like annotations into our annotation tags, and the GUI will just not harm them and preserve them, but not show them. and the simulator does ignore it. So
Yeah.
that is something that could do easily.
Yeah, it looks like CompuCell ignores anything between the annotation tags too. Just doesn't, the parser just ignores it. And so what I was asking is if I'm allowed to just put an XML annotation pair, tag pair in a comment in Python, and then Biomodels is able to look at the Python as just text and ignore everything that's not between a tag pair. The only things that'll be tagged will be the annotation.
Yeah, that's fine. I mean, you can do that. I think then what we need to do is to write, write code that can pass this and get the information from the Python code. That's what we need to do to get the information.
My guess is your parser may do that anyway. In other words, your parser may ignore everything that's not in a tag pair already, so you don't have to do anything.
Maybe not. I don't know. We need to ask them. It doesn't matter. It's a minor thing. If it does, then we can change it and ask, okay, for this format, do it this way. If it's a Python, then do this. We can do the other way. So we can just specify that. That's not a big problem.
I mean, so in that case, all we have to do is say that you should put a comment with open annotate, close it to annotate tags, and then whatever it is that we decide to put inside of those in the Python. And then it's up to you to parse the Python and throw out everything that's actually Python.
That is fine. Fair enough. If you can.
Now it would be nicer if we had a right clicker that opened up dialog boxes to make this happen. But at the moment, as far as the standard goes, if it's in a comment, we don't care.
Yeah, exactly. Yeah.
As long as you can handle the fact that there's free text that isn't anything that's meaningful to you, you don't care about that. So that makes it very easy. We could already put it in the XML. It works as far as I can tell. Maybe I put it in the wrong place, it'll crash it, but it looks like it works. I tried putting annotate tag pairs in a bunch of places, and it does the code runs. It just ignores.
But the disadvantage of that is that you get the annotation, but you don't know what it annotates if you extract it.
Well, that's going to be the problem we're going to face repeatedly, because the object, but even with what you had, what you showed, it's not so clear what is being annotated.
Exactly. Yeah, we know this term. We don't know what this means yet.
Because
in that case, of course. But if you have an annotation tag, the parent is annotated. That is just a clear relation.
Well,
we have this relationship objects as well. I mean, it's in the tree. It knows where it came from.
No, but find one of the annotates in your example, Jörn.
If you scroll down, it's a
not
field.
so obvious what's being annotated.
No, these are top level. But if you scroll down to the fields, it's a field a bit up, and 3 lines down. Here, exactly.
This one.
So you have field. The symbol is psmat5, or in this way, it's M mouse BMP4.
Yeah, before, and then you had diffusion for that, and then you have annotation for that protein.
So you would need the field information together with the annotation.
If we click on that, it goes to BMP4 mouse. Yeah, okay. Go back. So here, this is BMP4 mouse. I think you have this information in the name. Okay.
I think it's like you do it in SBML with the species, that you then take the symbol of the species together with the annotation from that species. That pairing could be done here as well, just that we don't have the relationship qualifier yet, but we could also add this when we have the full RDF back, then we could also have this relationship.
Yeah. Okay. So one problem I see here is the way we do this is because we can pass an SBML. We know what the field is. I mean, we do not know. I mean, here what this field.
It's not a species. It's not a species. Yes.
Not species exactly. Yeah, the thing. So because we don't know biomodels know this format, it will not know what field is. So if you read if it reads this BML, it will know what this if when it gets a reader tag called species, know this is a species, and then it will look for annotation within that, and then it will automatically connect the species with annotation. So it will know the name of the species, it will read and then it will understand the better. But this is a new format, and then it asking biomodels to read this, understand this annotation is connected to this field. That is going to be tricky because then it needs to understand the whole specification itself. So that's why in this case we need, if you have a common specification such as MultiCellML, which is working in both, then we could basically ask biomodels to read that and then connect the component with annotation. But that is going to be a bit tricky to do it for every format. If you want to do it for CC3D or Morpheus ML and then tissue forge and whatnot, and then basically it will be quite a bit of work to ask to read everything. But if you say we don't care and then we just get the annotation to make it more searchable, then it's fine. That's easy. But if you want to make the annotation connect to the component and search based on that, so if someone wants to search, give me all the models where the field is BMP4 or something like that, then that is not possible. That is
you could just ask with models which contain BMP4 is all right.
Yeah, that is possible. Yeah, that is
because even within a Morpheus model or a CompuCell model or a model using Vivarium or otherwise, the same chemical species, for example, could be represented in different ways. It could be represented as a bunch of concentrations inside of cells, in which case it would be nodes and ODEs. It could be it could be a chemical field. It could be other kinds of objects. And so the important thing would be that there is an object called BMP4. Now, there also is generally a shared concept of a field in all of these systems. And you might say, well, we know that we have enough of a field that we want to say by default the field has certain attributes. It's always, it's always going to have boundary conditions. It's always, which probably you don't need to annotate so much. Again, it depends a little bit if you're using this to try to actually provide a platform independent model specification, which is hard, or whether you just want to make it searchable. If your goal is searchability, then you don't care quite so much. In other words, it wouldn't be hard for us to agree that fields have diffusion constants and decay constants and things like this. Although those could get pretty complicated, right? CompuCell and probably many of these other things, we can have spatially varying diffusion constants. Constant information can be carried by cells. So those are not things that are easy to, that level of flexibility means that annotation, coming up with a fixed annotation standard is difficult. But I'm saying that the field has to have a name, and what kind of field it is not a bad thing. Chemical field, electrical field, force field, velocity field, those would be reasonable tag annotation.
That will be possible. So other just to comment here, I don't want things to be done this way. So if you're having a Python code and then if you add this hash and putting your comment, you can add annotation back, or you can add a meta ID at every place, and then in a separate document, then for each meta ID, then you have annotations. So then your code will be clean. It will have certain meta IDs at different locations, and then annotation will be in a different file. Then you need to pass this, or if you don't care about searchability, then you just pass the annotation file and you put them together. But if you want to connect them, then you need to get the meta ID and then link this with annotation, which is in the different file.
You probably want to be able to do both. Right? You want to support both of those options. Inline annotation and basically include annotation by reference.
Yes. Yeah, I mean the possibility. I think it depends if you guys all agree to do this one way or the other way, or both.
I think we will go for this annotation tag because at the time when we build a model, then we know exactly what the components are, and it's then not so much extra effort to bring these links in.
Yeah.
Except if later in a year we agree on how to do it and then have to think again what actually was PSMAT 5 in this case. Is it mouse or human? Or then we have to think again much more. So we will probably store the knowledge that we have at the moment for the time being in this annotation tag, and then later rather convert this and expand the structure.
Yeah.
What if you don't have the appropriate annotation yet, just register it, or is that what the inline annotations are for, like that?
I mean, we could say at least we need like the CBO ontology that is already out there as an additional main resource, because Sharif, you had shown your PDF document with the instructions for annotating in the beginning.
Yes.
Showing reactions and species and so on. So I think we will immediately go to this plus the CBO, and I think it doesn't have a link through the identifiers resolver, but it will directly use the link that is under the ID. In the same bioportal that you have been using.
If it is not that, that's not a problem. If you tell us, then I can get this into identifiers.org. That's doable. So if you say these ontologies should be should be the list of ontologies we will be using for our multicellular models, then I can get them into identifiers.org.
So one such annotation I posted in chat that we will probably often use for cell division, where you have to specify whether it's a symmetric or an asymmetric division. And that's very nicely laid out in the cell behavior ontology already.
Okay.
And if you open that page, it has this ID on the right tab, and probably that corresponds to the identifier link that you have been using.
But this should be somewhere.
No, you see it on the screen. ID, and then you have this HTTP, CBO, biocomplexity, Indiana, and so on.
Yeah, okay, yeah.
So I see it plain text in the center of your screen, details, and then it's the 1, 2, 3, 4th line in the details tab.
Oh, this one, yes.
I think this would be copy pasted into the annotation.
Yeah.
When we have a piece of code that says, how do I divide my cell, then I will annotate it with this one.
Okay, this takes you to the entire ontology. And then it takes you to this particular thing inside it. Okay.
It goes directly to symmetric cell division oriented cleavage plane, right?
No, yeah, it says that, but it's going to go to this page. Ideally, we would want that to go to this page.
So maybe the ID has to be changed a little.
Yes. It's basically taking you to the OWL file, the ontology of the OWL file, and then it's saying this is the keyword you need to look within the OWL file. You need to have an identifier for each of these. Instead of having this entire thing, I mean, they're using this whole thing as an identifier. You need to have a specific identifier. It can be CBO:00456, some number, some sort of identifier for each of these.
And this is not automatically generated by the bioportal?
No, I think it depends on the OWL file. So the OWL file which you provide to the bioportal should contain this information, I guess. This one doesn't have one. Or I need to check a bit more carefully.
Not now, but I think if you can look into exactly this page and then tell us how we could construct this link.
Yeah, I can do that. I think.
Then we will use many of these type of links all across the model.
Yeah. Okay. Yeah. So the So this is from, so James, this is from you guys, isn't it?
Yeah, this is something, I mean, the problem is that this was a project that stopped.
I see.
And so I mean, if we had somebody to work on it, no, basically it's like a Wikipedia. This is a stub, right? We created a demonstration of it, but there's a lot to do still. Luca's the one. You will point things out to Jim. He will fix them.
Okay. So I think, from my understanding, it doesn't have an ID. If I go to anything else,
see if anything else has an ID, because I thought some of these had oboe IDs. So then that should work. Is it the fact that it's an OWL formatted that is the problem?
No, I think that's OWL format is correct. So it uses this as an ID, the whole term.
Instead of giving it a unique number.
Identifier, unique number identifier. But this,
just pull another, just go to some other let's see if they're all like that or if it's just that one.
okay, let me choose something else from BioPortal.
No, just for CBO.
Go to classes and then
choose some other Cs. Okay, that's fine. I can do that. My feeling is that they're all.
I know Go will do it properly.
Okay, let me just do that. Yeah, so go choose something else, like cell death, for example.
Yeah, just try cell death, see if it pulls up.
Yeah. See, okay, so it's the
no,
same.
It does it the same way in all of them.
Same way, yeah. It says the thing, and instead of identifier, it just uses apoptosis itself as identifier.
Right, but if you look below where it says is defined by, that's what you want. Those are the ones you want. That's what you want it to look like.
Oh, it has a spato. Okay, so you are taking from, this is taken from another ontology. So you're having that.
But what you want is, instead of ID looking like that, you want the ID to look like the ones at the bottom that you just highlighted.
Would that just be CBO colon?
Could be CBO colon number. Yeah.
Or apoptosis or your number.
Yeah.
Sorry.
and that is existing for some of the entries.
Yeah,
But
I think
not
these
for
are,
the. If you go back to the mitosis symmetric oriented cleavage plane, the third one of those, that's the one that we often want to tag, and that doesn't have this relationship.
okay. I tell you why, because that one is from Pato. Let's see, OLS.
So one of the problems we faced when we started doing CBO was that 50% or 60% of the things that are most basic in biology don't exist in any ontology.
Okay.
Even simple things like I can't begin to describe the most basic things that everyone needs all the time are not there anywhere, really nowhere. And so a lot of things you'd say, how can there not be a definition of this? The answer is there isn't. I mean, I see Aaron nodding his head because I'm sure you felt the frustration. And so the whole point of CBO was to try to fill some of that stuff in, but then we didn't have money. We ran out of money, and so it didn't get finished. If we had that, it would be great to spend a week of grouping people, of volunteers, building it, because it's fragments. But this is the annotation.
So we should be able to register new terms as you build your model and discover that things don't have proper ontology terms. You know, if you need a chemotaxis, you're looking through the ontologies and you can't find it. Like what's the process in which you just add it, get the number, reference it, and now it's there. I mean, it seems like these things come up all the time as you're modeling, but it'd be nice to do them as needed, maybe.
The problem is that the ontology editors are really unfriendly. I mean, those OWL editors are, yuck.
So there's a barrier there. As soon as you want to do it, you're hit the wall.
Yeah, I mean, it's not impossible, but it's not just, hey, type the term and get a UUID for it.
It's also
Because you
very
have to
important.
define all the
Yeah.
relationships and the structure and the structures.
I just shared with you on an editor, nice web editor for ontology. So I don't know if you know that. I haven't used it enough, but I think the ontology guys from EBI, they use this one. This is a nice editor for the ontologies. I think you can get. I don't know how fully it works. I haven't fully tested it, but I think my understanding you can somehow get in the old file and then add different branches, edit different branches, and so on. Then multiple people collaborate and work on the same ontology. Using
That's
this.
pretty neat. So maybe we should distribute that to everybody, send out that info out as a link in email, because that would be really nice to know about. I mean, you'll still have the same problem, which is it's going to be like you need to basically you need a Git repository, because you don't want people just crashing your ontology. So you have to have some kind of branch and review structure for it, which I assume maybe this editor has.
I hope so.
I mean, when we did CBO, I mean, it's already what, 12, 14 years ago. So I'm sure the tools are better now than they were then.
Sure.
You're right that one of the problems is someone registers a new term and it's trash. Now you're forever, you know. I mean, I guess you could just register another term that's better, but
Well, people do clean things up. I mean, if you look at Go was a mess and is still a mess to some extent, because they started at the leaves and went up. And they're very good. If you point out that something's at the wrong level of hierarchy and things, they'll fix it. So I think that it's not impossible.
You need someone to be reviewing those decisions and responding. So
You sort of need a curator for the ontology, and that's a big job.
Yeah, that's a big job.
But yeah, I mean, as I say around, I mean, somebody needs to do this. The question is, how do you pay somebody to do it? Is the problem.
Yeah. If people could self-register and there's just someone to review, like, you know, like a Git, you know, just you need to approve it before it.
Right. Again, the term definition is not so bad. The problem is the relationships.
Mm-hmm.
If you're building a structured vocabulary, you say, I need a term that means this. Adding that is not so bad, but then where does it go in your hierarchy? What are its other relationships?
Mm-hmm.
That becomes harder. I'm not saying we shouldn't. It's already good. You know, already just being able to register new terms and make them clear is already a big deal. But I'm just saying it's not the end of it.
Yeah. Okay.
It seems pretty necessary, and actually, like, the least painful way forward is to have just.
Yeah. I think James, it's a part of FAIRSPACE. We need to spend some time probably to get this thing cleaned and updated. This means some of us just work on it over a week or so, just to do some hackathon or something like that to specifically get this cleaned.
And also, yeah, to get that kind of thing funded is, of course, very, very hard.
Might be easier in Europe than the US.
Yeah. Alexia support these sort of things, I guess. I don't know. Did you apply for the Alexia EOS call? No.
No, too small a network yet. We needed more partners across Europe, Eastern Europe especially, but we don't have.
I mean, EOS, the Alexia, those sort of they support these sort of things. If you want to extend ontology and whatnot, maybe this can be also a nice project proposal for the Alexia biohackathon, where you can propose this, and then there'll be more people coming to a very nice location, and they are fully paid event, so you can attract quite a few students or researchers to work on this for like one whole week. That would be really good.
I mean, my guess is that Jim Sluka, if you paid him to come, would be happy to take a week to do this. You know, if the travel were covered, but and because it's really his problem. We started it before he joined, but he's the one who delivered it. He's the one who knows what's there, and how he edited it and structured it.
You can see that it's from 2011, 24 August. Right. Okay.
But beyond extending it, James, I think it would already be a step to just register UIDs for all what is there already, even though you say it's a stump, but just preserving what we have already and being able to refer to it.
I agree. I don't know how to do that though.
Sharif, that's probably
Oh,
identifiers registry.
Yes, I can ask, and then maybe see if it could be done. Maybe get some help from the ontology team, because we have a very big ontology group to do this sort of things.
It might be of interest to the Reproducibility Center.
Yeah,
but more focused on biochemical, but the next cycle we want to introduce more multi-cell level ontologies. The annotation person within Reproducibility Center is John Gennari. I'll talk with him.
yeah. If anyone volunteers to do that, it would be great. We need new caretaker for the CBO.
But still, until then, would we still use the ID with the OWL link and then later transpose this into an UID?
Yeah.
If you want to store some knowledge that we have at the moment.
It's still a unique identifier. It's just not a convenient one.
Yeah, exactly. Yeah, that's correct. Yeah, that is still unique, right? It's not convenient. Yeah.
So when this is submitted to biomodels, you will then have a script later to go through the Morpheus ML files in biomodels and then replace the identifiers.
I don't know. I mean, there are several ontologies we don't even resolve because it's not easy. They don't have a proper API. I mean, if things like this is being added and then there's no API or proper support to resolve it, then we will just show that ID. It will look like a big. It will just show this link or something, which will look nice. So it should be resolvable at some point. And also, it's a big challenge, right? So if you want to resolve different ontologies, you have to use different APIs for different people, which is not easy. It's a pain. So we have, because we use this collection, which we know is the one which we mostly use. So we have to write custom code to resolve that. So if it's NCIT, it's going to use certain API to resolve NCIT. If it is Uniplot, we know we have to use Uniplot's API to resolve this. There's no single API which we can call to resolve everything. That is a challenge. So more different resources means more work internally for us.
Yeah.
Good. All right. So what should we do next? Maybe if you pick up a model, and if you add annotation, and then maybe add more annotation also to Lorentz model, a CC3D model, Lorenz's model, we can, if you give it to us, then we can see what can be done with that annotation. If you can make them indexable, findable.
Pedro or Lorenzo, do you want to try that?
Yeah, we can try.
I mean, I'm not sure how we should we have a breakout room or something? I mean, I'm not sure where we do it. In other words, it's not obvious. Do you want to do that in public in front of everybody, or shall we So how do you want to do this?
So we will do it over the coming weeks and probably step out in a
Okay.
minute from
So
here.
you didn't meet it right now. I thought Sharif was saying to try right now. Was my understanding of what he said.
Yeah, maybe later tomorrow or something for people from the Europe, and then just a few example, and then bring it for the discussion. It's fine.
Maybe it's worth opening. Pedro, if Pedro can screen share, why doesn't he open one of his models? And we could just talk through the principles of it, and then he could try doing it a little bit and then present what he got tomorrow.
Yeah.
Okay. I just need
Screen share privileges.
yes, I don't know who's sharing. Rahman
Sharif,
needs
I think.
to
Stop it. Yeah. Okay. I'm just stopping it. Sorry.
finish.
And Lutz, you could give us some guidance here about what sort of principles should be. So it looks like, okay, so the first thing is, so we could use this annotation and annotation tag anywhere. Where should we be putting them? This is our XML. Not so
We can
different
do-
from yours, of course.
We can do this. I mean, and
So-
type something here. Like
so
this.
Like this, we also have it in the tree. Yes.
So where does your model start? So from the top, if you go, metadata model.
So at the very top, you could put in all the all the information about author, name, user ID, references, et cetera, right?
The PubMed link.
No,
Yeah.
you can put them all within the annotation. Using the V cards.
No, you
Within-
need to use the annotation tags.
Yeah, so that's fine. Instead of going into the plugin, exactly, somewhere.
Just after, no, I wouldn't put it there. I'd put it, it should be inside, right? It should be inside of the CompuCell tag, shouldn't it?
Yeah. On line 8 and following.
Yeah, exactly. Yeah,
It's got to be inside the object it's describing.
and then as you know, how do I paste this? Can I paste some example to the
In
working
the
document?
chat. You can put it in the chat.
This
Yeah, that's fine. Yeah, I mean, but can you copy? Okay, let me just get that for you from the chat.
But you have all the tags, right? Author, journal
Yeah.
article, abstract, keywords, all of that stuff should go there.
Yeah, okay, let me just share something in the chat. Give me a sec.
Now it could be put into a separate file, but it should be, can go there too. Just an example of what needs to be there.
Okay, for example, this is our taxonomy. Can you just copy that? Yeah, that has to be within the. Yeah, I mean, yeah, just remind first cell copy cell. Yeah, that's cool. I think this is good.
Okay, now what do we do with situations where we don't have tag pairs, but we have a short form tag? Like dimensions, right? We don't have open dimensions, closed dimensions tags. So then we can't put an annotation tag pair inside of that.
Yeah,
you can do this.
Presumably those can be extended, but I don't remember how to do that.
And then
But you
here,
Cool.
That's what you need? Okay, fine. I just never
I
remember the
think-
syntax. Yeah.
I think this works. Let's
Okay.
try.
Great. If that doesn't crash, then you're good.
It works.
So you can put, so in any object tag, you can put an annotation. Okay, good. So now you have to decide what we want to annotate and how, but you can do it. Okay, so now let's look at the Python. So I think in the Python, we're just going to say you're gonna, wherever it is, after the object that you're annotating, in line with the object or in the line afterwards, you have to decide. You're gonna have hash and then open annotate, close annotate.
Okay.
And then you just use exactly what you had there. Except you're not going to be able, yeah, you have to put triple quotes. Yeah.
Does it make the code a bit ugly?
It will. And at some point we probably will want to make it, we'll make it
Well, I know it's going to be a pain, but would you consider looking at the way Tellurium does it?
how do they do it?
I forgot there's some way.
Don't they use an @ sign or something and then
Yeah.
they have a decorator? I don't remember the decorator. But yeah, I think long run you want to have something like hash @ something, and then that's what, and then that references the thing in another file.
I mean, even if you say annotation, I mean just hash and then something like BQ by all has taxon or something, then space this link, and then that's all what we need to know to get this.
Yeah.
You don't even have to write annotation. Or, I don't know.
Yeah, either one of them, but that's legal Python code. And I believe your parsers will just ignore everything that's except for it's not in a tag, so you'll be fine.
Yeah. Okay, yeah, so
The second one would be nicer, but we have to decide what the format for that link is, so that it, and then in the other thing you have to have something that corresponds to it.
Yeah.
And then I guess, Pedro, if you want to do this, then you need to go to the bio portals and try to find some reasonable references for things. And again, as Aaron was saying, a lot of the stuff you want won't be there anywhere.
I have a question. How, for example, when I submit the simulation, or which software will scan my simulation for annotations? When does that happen?
So I think when the biomodels team is curating your upload, then they will parse and prepare the metadata for the web page.
Okay. Okay.
So that's why we want to try this, because we want to have something where Sharif can try parsing it and seeing what happens when he does. But I think the basic idea here is reasonable. The only thing that you have to do is you have to go to bioportals and then start digging for ontology references.
Yeah, or OLS.
Yeah, or OLS. If you want OLS, then send him give him the link that you want him to use for his searches.
Yeah.
And I get, we don't have to annotate absolutely everything. We just want to have something that's illustrative so that we can see what happens when we do it.
Yeah, that's fine. Share some example. You just share the OLS link here as well.
But I think just getting authors, title, abstract, and keywords into these things would already be a lot.
Yep. Yeah, so last link is the OLS. So here you can search for any keywords and then get this term.
Persistent motion.
Yeah.
Well, metabolism it'll have. How about persistent motion? Bet not.
Let me just okay. There's a more complex definition than that, but never mind.
You don't care. You don't care. Just make sure it's not wrong. Persistent motion. T. That's one. Persistent motion. Nope. Not there.
Not there. I think that's why we need CBO to be fixed. Yeah.
Well, that's all right.
Migration. Yeah.
I think it's more biological than physical, right? In the sense that
Yeah, this is more
consistent
Physics is a
migration.
problem. Try cell migration.
cell migration would be there.
Cell motility, probably it has stuff. But again, it tends to be, most of the things you need are not going to be there.
Yeah.
And then it'll have super detailed things that are, you know, it'll jump down to these very, very fine grained things that are not.
Okay, so motility.
Oh, so there's so cell motility you got. Yeah. Go and NSIT are usually pretty good. I mean, look at that. Cell motility involved in camera eye type morphogenesis. What the hell? That's why I say Go is a mess.
I think we got only 2 minutes before the next session starts. So
Penkai will need the room, yeah.
Yeah.
yes.
But I think explore this a bit, Pedro, because I think what you learned, what you learned from your successes and failures will be very informative for tomorrow.
Okay, so I try to annotate that simulation. Okay.
Poor
And,
Pedro.
you can also add few annotations for the Morpheus, one of the small models. I mean, it doesn't have to be fully annotated, like just a few of them here and there. And then we can ask them to test it out to see if that works.
Cool.
Okay. Okay. Fine.
Thanks, Pedro. That's great.
Thank you.